Yeah.
All right. So we have 3 repeated spaces in the cochlea. A scala tympani containing perilymph, a scala vestibuli containing perilymph, and it's also where you find the vestibular membrane. And between them, a small endolymph-filled space called the scala media or cochlear
And that's
duct.
that big model right there.
So this set of 3 exactly matches this model here. So take a look at the small model. So this is the green, this is the blue, and this is the pink. Exactly, matches up these set of 3.
Which one? Okay,
right.
so that the blue one is the one that is.
That's where the depolarization occurs,
Okay.
where you get the depolarization of the cochlear branch axons, the release of the neurotransmitter, and so on. So the specifically sensory region of the cochlea would be all the blue spaces. Okay?
Okay.
All right. So, scala tympani containing perilymph, scala vestibuli containing perilymph, and it's also where you find the vestibular membrane. And then inside the scala media or cochlear duct, we've got a structure called the spiral organ or the organ of Cordy in here. It rests on a membrane shown in white, the basilar membrane. It's got a roof-like membrane over it called the tectorial membrane, and there's a group of hair cells in between. So hair cell ending with the hairs, hair cell with hairs, hair cell with hairs, hair cell with hairs. Now these hairs act like dendrites. The hair cells are not actual neurons. No, they're actually excitable epithelial cells. Who knew that epithelial cells could depolarize and secrete neurotransmitter? It was a big surprise when that was discovered. But these hairs, which are actually called stereocilia, act like dendrites. They are responding to a mechanical stimulus. And when they do, they will depolarize and secrete a neurotransmitter compound. So what would actually happen would be something like this. Something over to here, so we would have sound waves funneled by the outer ear or external ear into the tympanic membrane right there. And the tympanic membrane or eardrum goes in, out. Every time sound waves hit, it goes inward. And you can see, okay, this is broken. That's not good. Okay, let's see if we've got. It's also broken. Not good. Not broken. There we go. Not broken. All right. Put this back in. All right, so sound waves will be trapped by the auricle of the external ear, funneled in. So what is this, by the way? What would this space in here be?
External auditory canal?
Canal, yes, external auditory canal. Yeah, exactly. So the sound waves that are funneled in strike the lateral surface of the tympanic membrane. And the medial surface of the tympanic membrane, we have, actually there should be 3 middle ear bones or ossicles. We have the malleus right there, the larger incus, and there should be fused to that a stapes. So in other words, they've separated the stapes. That's an error on the model. Those three bones fuse together right there. They should fuse together. And my point being that when sound waves strike the tympanic membrane, it bows inward, and these 3 bones move inward also. And the stapes starts to tap, tap, tap on the vestibule, specifically tapping on the oval window here, pushing it in. Now, liquids are non-compressible. Liquids are no more compressible than solids are. So in order to be able to push inward to compress the liquid, you've got to be able to have, to release that pressure. So the role of the round window is, so every time the stapes strikes the oval window, the round window bulges out. That allows the oval window to get pushed in. When it gets pushed in, those liquids, the perianum, will begin to move back and forth. So here, the endolymph is moving back and forth in here. The hair cells bend one way, ion channels close. The liquid moves back the other way. The hair cells bend the other way, the ion channel is open, and you get depolarization of the hair cells. They begin to secrete neurotransmitter glutamate onto the pseudo-unipolar neurons shown in yellow. There are those cell bodies, excitatory receptors to the glutamate. We get depolarization to threshold, action potential traveling along these cochlear branch axons here. You also see the vestibular branch axons coming in, obviously from the vestibule and semicervical canals. Here we see them merging to form cranial nerve 8 and vestibular cochlear nerve. Okay, so action potentials for the detection of sound coming along here, action potentials for the sense of equilibrium coming along here. Okay, that help? Or was that overwhelming?
A little both.
A little, okay, helpful, and you're going to need to mull it over a little.
Yeah.
All right, got it.